Hello everyone. So today we will be discussing about gram-positive cocci. We will briefly look into what gram-positive cocci are, and after that we will see how can we identify them and classify them based on their shapes and appearance under a microscope. We will be doing some exercises by looking a few slides under microscope and identifying the organism and telling how we can describe these organisms. So let's start. So what does gram-positive cocci means? Gram-positive, as we all know, there is a gram staining which is performed on bacteria, and on the basis of the staining results, we call them as either gram-positive or gram-negative. If after gram staining the bacteria appears purple colored, we call them as gram-positive. If they appear pink colored, they are called as gram-negative bacteria. Now to get into the details of this gram staining, I have already uploaded a video on gram staining. You can watch that. But briefly, if the cell wall of the bacteria has a very thick peptidoglycan layer, then it retains the primary stain of the gram stain, which is crystal violet, and it is violet in color. So when we decolorize them or wash the bacteria, it is not washed off and it retains this purple color and appear purple under a microscope. So these organisms are called gram-positive organisms because of the structure of their cell wall. Now you understand this cocci word. Cocci means spherical or round. So gram-positive on the basis of gram staining and cocci because they are round-shaped bacteria. We call them as gram-positive cocci. So they will basically appear as purple-colored balls under a microscope. So cocci are always round and spherical in shape. This is now clear. Now how they divide and arrange themselves after division is the key to their identification. All these bacteria divide to multiply. In what plane they multiply will determine that how they will appear under a microscope. So when you look under a microscope, if these bacteria appear in the form of clusters or a bunch of grapes, you call them as staphylococcus. If they appear in the form of chains, that is, various bacteria are lying next to each other forming a thin chain, then they may be most likely streptococcus or enterococcus. If we see bacteria as a pair of 2 gram-positive cocci, there are two bacteria, they are appearing in pairs, we call them as pneumococci. Whereas in case the bacteria are appearing in the form of a tetrad, where they are present in a square of 4 cocci side by side, then we call them as micrococcus. So this is the appearance, what appears under the microscope. In clusters, usually staphylococcus appear, short or long chains. You will see in case of a streptococcus. In a pair of 2, you see in case of a pneumococcus. And as a tetrad, you can see in case of a micrococcus. So staphylococcus, they appear as grape-like clusters. They are divided in multiple planes because of which they appear as bunches of grapes or clusters. Now, if we perform catalase test, these organisms are catalase positive. That means they form bubbles when the colonies are touched to H2O2 solution. Common organisms in this group include Staphylococcus aureus and the other category which we call as cons. Cons include Staphylococcus epidermidis and Staphylococcus saprophyticus. So, in short, you can remember that Staphylococci appear in the form of clusters. Common organisms are Staph aureus and coagulase negative staph, that is epidermidis and saprophyticus. Now, what diseases these cause? They usually cause pus formation in human beings. So, these pockets of pus are called abscess. So, they cause abscess or pus forming infections. Coming to the streptococcus. So streptococcus mainly appears in the form of chains, long chains of bacteria because they divide in a single plane. They divide in one plane because of which they will appear as different cocci that are lying next to each other. And as you remember, catalase positive was staphylococcus. In case of streptococcus, you can differentiate because it is catalase negative. The common organisms in this category are Streptococcus pyogenes. It is a common cause of sore throat. Another is Streptococcus pneumoniae. Streptococcus pneumoniae is a common cause of pneumonia, as the name suggests. Now, in case of pneumonia, these are usually arranged in a pair of 2 cocci. When there is pair from P, you can remember pneumonia. And if there are chains, then it is Streptococcus pyogenes. Enterococci are very similar to streptococci. They appear as either pairs or short chains. Usually the chains of streptococci are way longer. but they also appear similar to streptococci because the cocci divide in one plane. So the cocci will be lying next to each other, and just like streptococcus, they are also catalase negative. Common species in this category is Enterococcus faecalis and Enterococcus faecium. These are the two most important categories or most important species that cause human infections. What kind of infections they can cause? They can cause urinary tract infections, endocarditis, and some abdominal infections also. Now, micrococcus. Micrococcus is not very important clinically because they are usually non-pathogenic. These are environmental bacteria. What happens? They appear in the form of a tetrad. Tetrad means four bacteria lying next to each other to form a square-shaped appearance. Okay, so these are usually non-pathogenic. The common organism in this category is Micrococcus luteus, but it is usually an environmental bacteria. So for now you have seen classification. First of all, we will do gram stain and classify if it is purple or pink. If it is purple, we call them gram positive. If it is a spherical or rod, if it is spherical, we call them cocci. So they are gram positive cocci. Then we look at the arrangement after they have divided. Either it is cluster, we call it staph. If it is chain, we call it streptococcus. If it is pairs, we call it pneumococcus. If it is in tetrad, we call it micrococcus. And on performing the catalase test to confirm, we get positive catalase test in staphylococcus and negative in case of streptococcus. So this is a flow chart showing first you will be doing gram staining, gram positive, gram negative, cocci, bacilli, clusters, pairs, and chains, pairs, and tetrad. So this is a simple classification of gram positive cocci. Now we will do some exercise. You will have to identify the organism. First, you will tell what is the description, like this organism is this, arranged in such and such condition. So identification, most likely this is species. This is an actual picture of the organism, how it appears under microscope. So you can see there are long chains. Okay. So the description will be gram positive cocci in chains. Identification, which organism is this? This is most likely Streptococcus species. The next one, you can describe it as gram-positive cocci arranged in mostly clusters. So which organism is this? It could be Staphylococcus species. Now these are gram-positive cocci appearing in pairs. You can see here they are appearing in pairs. Okay, there are two cocci lying next to each other. If you see minutely, there is a little halo also around the bacteria. Here you can see a little halo. So this halo is actually capsule because pneumococcus is a capsulated bacteria. Here also you can appreciate a capsule. So this is Streptococcus pneumoniae or pneumococcus. Thank you very much for watching the video.